Thanks for inviting me to show our recent work. I changed my title a little bit. I'm gonna finish within 10 minutes. I will go through all slides. I include some other work that we are working on. So the title is Data-Driven Functional Analysis of the Human Virome. So now I'm an associate professor in Tsinghua University in China. This is the top one university in China. And our lab working on human virome. So everyone knows like human virome is ubiquitous, very diverse and very large. The number of the particles, the viral-like particles, is similar to the bacterial cell number, and they are quite diverse and dynamic. So, for example, when we talk about virome, like most of the time we are meaning like eukaryotic virus, prokaryotic virus. We are familiar with a lot of eukaryotic viruses. They call it infection. For the phages, actually they are the major part in the human virome, especially in the gut virome. And about more than 10 years ago, the like people found that the phage population structure changed in disease status. Like so far, like most of the population structure are association studies. So for the causality mechanisms, we don't know. In recent years, people start to do fecal virome transplantation, and most of them in animal models. But seems like the fecal virome transplantation can also change the disease phenotype. Most of the time, like if you change the fecal virome to a disease model, and it can recover the disease. Again, like most of the study, like we don't know the mechanism. So I would say like in the human virome study, especially related to human disease, there are two major challenges. First, like we know the identification, dark matter identification, new viruses, new functional genes. Another is the mechanism, the gene, the functions. So what's the mechanism of the dynamics of the virome in disease, and what's the mechanism of the fecal virome transplantation? We don't know. So our group, like we focus on these topics. We design different experiment and analytic tools for those kind of studies. The main one of our focus, like Jeremy just introduced a little bit. I'm not going deep to do this. We focus on bacterial prophages because we found like in our previous publication, we found, and also other publications in animals, the bacterial prophage are dominant reservoirs of the human virome. Like a lot of the bacteria in our human guts, they contain prophage, and most of them may be induced. Okay, so we have a long-term hypothesis that like prophage may contribute to shape the gut microbiome and to impact the human health. One hypothesis is that prophage can carry some genes, and those genes can modify bacterial fitness and change the virulence of that bacteria and affect human health. That's our hypothesis. To figure out this hypothesis, we started to work on some projects. First one is like we want to design some tools to identify prophages. And we know like we already have some tools available, but we want to introduce in the deep learning method, especially the language model, large language model for those kind of identification. Last year we published a paper like we introduced protein large language model ESM2 for prophage identification. So how did we do it? We use ESM, the large language model, to help us to distinguish prophage of and bacterial gene and bacteria of. For example, like if this large language model can distinguish these phage proteins, and we can go back to the bacterial genome, and we can identify the location of each phage genes. And then we calculate the phage gene density, and we can find high confidence prophage island. Like those regions may be prophage island. And those are the algorithm that we use for phage gene density calculation. And we found like we did some like benchmarking for wet lab validation, we use the metamethylin C to induce some bacterial prophage from the bacterial strain that we have in our lab, and we made a data set, and we did benchmarking for different tools. Like we cannot say like PAD is best, because like most of the time we don't know whether a prophage can be induced or not. But we can see PAD can recover most of the prophage that compared to each software that's widely used. And also we did a base level prediction for the prophage boundary. And interestingly, we found like a PAD can improve prophage boundary identification. This is very interesting because PAD was not designed for base level prediction. However, seems like the large language model can recognize the boundary phage proteins and in the genome. That's why it can perform very well in the boundary identification. So this tool can also use for virome data analysis, like it can be used for phage contig identification. And it's totally publicly online. Like we got very good comments from the reviewer when we submit the paper. So then we have a tool to identify prophage. Next, we want to know the function of those prophage encoding genes. We developed a tool called futC. It's a large language model based tool. It can identify whether a protein belongs to an enzyme or not, and also classify the enzyme specifically into different levels. So, and you can see that like the futC can perform very well on binary and multiple classification tasks for the enzyme. And also it did very well on low similarity sequence identification. Similarly, it can identify real dark matters. And based on this algorithm, we developed an end-to-end pipeline for metagenomic enzyme functional profiling. And this pipeline can analyze data from raw data to enzyme functional enrichment. And it can also trace the enzyme genes to their bacterial host. Okay, so far we have and this one we have online platform like everyone can use this. It's totally free. So far we have the prophage identification tool, and we also have the functional annotation tool, and we integrate all these tools to profile prophages in the human gut microbiome. First, we use the UHGG database. It contains almost 300,000 max there, and we found that more than almost half of the bacterial genomes, they have prophage there. And the distribution of prophages among different bacterial traits and taxonomy are also identified here. Furthermore, like we expand the diversity of the gut prophages and also look at the features of those prophages. So we found a large amount of prophage cannot be annotated, even taxonomy and both taxonomy and functional. And then the predominant family identified were Padoviridae and Venoviridae. Like those are the new names after STV changed names. And some bacteriophylla seem to be preferentially integrated by certain family of phages. I'm not going into these features. And next, we annotate the function of those cargo genes of those prophages. The cargo genes are the genes that were not related to the structure and the life cycle genes of the phages. So we found, okay, those cargo genes, their functions really are quite diverse, related to many functions. And so above, we analyzed prophage in a database. Next, we did the analysis for the population level, and we collected 8,000 metagenomes data. We linked the prophage abundance to the population features. So it seems like the y-axis is prophage abundance. The prophage abundance relates to the age, gender, industrialization level, and the different countries, something like this. Okay. So interesting is that we identify the abundance of the prophage in different diseases. We found the ratio of the prophage were higher in the disease status of most disease type. And we also validated this in the mouse model. This can be explained by the diversity of the bacterial. Seem like because in most of the disease status, our bacterial density is our bacterial diversity is low, and the low bacterial diversity may link to prophage formation. Okay, so move on. We analyze the cargo gene function in different prophages in different disease. We found the dynamics of cargo genes functions is quite disease specific. For example, in IBD disease, we test 7 cohorts here. Most of the prophage encoding genes that relate to immune modulation, they were depleted. A lot of them they lost in the cohort. Okay, so next we did further analysis. We isolate. We identify one gene called futC. This is a prophage encoding the oligosaccharide biosynthesis genes, and this one is associated with IBD disease. It was depleted in the cohort. Okay, so we then we clone this gene and found this gene can synthesize 2FL is an oligosaccharide efficiently and with very high efficiency, and we feed the mouse with this product, and we can see that so this 2FL could trigger intestinal IgA secretion and intraepithelial CD4 CD8 positive double positive T cells in the mouse gut. Seem like the prophage encoding gene can synthesize some oligosaccharide, and it can modulate the human immune systems. And that's our general idea of this project. To summarize the project that I mentioned today, so we developed some AI tools that can identify prophage and prophage gene function. We expanded the prophage diversity in the human gut, and we did the landscape analysis. We discovered disease-specific cargo genes in prophages. And we performed the experimental validation and a little bit of mechanism insight into how the prophage encoded gene can modulate the host immune response. Thank all the students and the funding agency. Thank you. That's all I want to talk today.